next, we have a really exciting speaker joining us. We have Archana Anand, and Archana is an environmental microbiologist, and she primarily is working in the interface between environmental engineering and coastal marine biology. I'm going to bring you on in here so we can all see you. Hello, welcome to the Global
Hi,
Bio.
Lizzie. Thank you for having me.
Yeah, it's wonderful to have you aboard. So we also hear that you're intrigued by aquatic microbes that drive several vital processes in the land and the ocean. So we're really excited to have you on board with us today and hear a little bit more about your work.
Of course.
So
Likewise.
I'm going to bring in your screen share for us now.
I just started the slideshow. Can you see it?
Yes, it looks perfect.
Okay.
All right, I'll let you take it away from here. And if those of you who are on our live stream, please feel free to add questions into the chat and we can get those answered at the end.
Thank you, Lizzie. Well, hi, everyone. Welcome to The Path of a Flush. Sewage can tell stories. Before I begin, I want to thank the event organizer, fellow explorer and friend Joe, for inviting me. Now, can anyone recognize where this picture was taken? This is Marina Beach, very close to my childhood home. I was born and raised in Madras in India. I come from a family of teachers, and naturally, the passion for education was instilled in me from a very young age. I spent most of my adult life in Singapore and Hong Kong, and I've now been in the San Francisco Bay Area for about 2 years now. So, here's an overview of what I'd like to share with you today. First, I want to take you through my science journey from engineer to biologist. And next, I want to talk about how I spent the valuable years of the pandemic listening to stories from wastewater. And of course, what all that means for global biodiversity. I'm a mechanical engineer by training, and I graduated in 2007 with a degree from the National University of Singapore. I worked for a few years and traveled widely, but I wasn't very sure about what I was doing. And after a few visits to Africa, the Himalayas, this was with my partner and fiance at the time, I wanted to learn more about the environment and use education to conserve it. So I did the first thing that I thought might bring me closer to my goal, and I enrolled in a master's in environmental science at the University of Hong Kong. Now, this was where I met Professor David Baker. This is David on the floor. And I was intrigued by the work the Baker Lab was doing, looking at the impacts of human inputs on coral reef ecosystems. So we had a meeting, and I asked if I can do a project for my dissertation. David, who ended up becoming my PhD advisor, was skeptical because I was an engineer and he was a marine biologist. So we decided that a good starting point and middle ground would be chemistry. I was thrilled. The catch, I had to work on wastewater. The lab had just been successful getting some funding to look at nitrogen impacts on the marine environment. And they needed someone with a blend of quantitative skills and marine biology, basically me, to look into this and interest in these fields. And so it began. Now, let me give you some context on why all this matters. The United Nations Environment Program estimates that over 8 million tons of marine litter reaches the oceans every year. Now, just 10 rivers carry over 90% of the global litter into the oceans. 8 of these rivers are in Asia. Everyone is talking about plastic. We've banned the use of plastic straws. This is great. But what about sewage? Why aren't we talking about sewage? Do you know where your sewage goes? Sewage is rich in nutrients, such as nitrogen and phosphorus. Now, nutrients are important, but when in excess, they can accelerate the growth of some microbes, and these microbes begin to respire rapidly and deplete oxygen concentrations in the ocean. This can have detrimental cascading effects on the life and the health of the ocean. Hong Kong is no exception to this. The city has a population density of over 7,000 people per square kilometer of land. Compare this with, say, Iceland, which has a population density of only 3 people per square kilometer of land. In fact, Hong Kong dumps over 1,000 Olympic sized swimming pools, equivalent of treated sewage, into the ocean every day. Consequently, the impact of urbanization on the marine environment is stark. But why does this interest me? The reason this interests me is that despite all these environmental stresses, the marine environment is rich in life. Did you know that Hong Kong has many more hard corals than you can find even in the Caribbean? So, what was the big question that kept me up at night? How does nitrogen pollution affect the ocean? And I split this big question into two smaller research questions. How does it affect life? And how does it affect health? Now, this was also when I got an amazing opportunity to lead a large collaborative effort with research teams from the University of Hong Kong and the Smithsonian Institution in the United States. But for all this to materialize, I needed an important skill: scuba diving. Now, this was hard work, but I got certified in open water, advanced open water, rescue diving, and scientific diving. And to date, these have been some of my best lived experiences. Now, coming back to the project itself, did you know that over 60% of life in the ocean is either unknown, unnamed, or invisible to the naked eye, and yet contributes to majority of the nutrient cycling on Earth? So to study these tiny critters, I used structures that were called autonomous reef monitoring structures, ARMS, or mini ocean hotels. And these mini ocean condominiums or hotels are about 9 inches in height. They are made of PVC plastic. They have gaps in between for water to flow through freely. And I scuba dive. I deploy these in the ocean floor for about 1 to 2 years, after which I collect and identify everything that has colonized the plates using photography as well as their DNA signatures. Now, here's a video of me with my dive buddy Mike in Hawaii retrieving an ocean arms. And you can see we've placed a white bucket, secured it with bungee cords on the arms. And here on the right, you can also see another unit that's still soaking in the water while we retrieve this one on the left. The reason we're securing it with a white bucket is because we are interested in not just what is on the arms, but also what is in the water that is immediately surrounding the arms plates as well. Now, arms retrieval and processing by itself is a huge team effort because it allows for the involvement of so many students. I think my screen is frozen. Oh, okay, yeah. So you can have many students being involved, and not just students, but also the general public as well. So here I want to show a video of our lab retrieving the ocean arms and processing them in a huge citizen science effort where we're trying to build a genetic library of Hong Kong's marine environment. I hope you enjoyed that. This is one of my all time favorite slides. It shows some of the organisms that we found on the arms from Port Island in Hong Kong. And interestingly, we found a higher abundance of pathogens in sites that had more nitrogen pollution. And we're still working through the data, and we have a lot of interesting things to report as well. Now, around this time was also when I had my first baby. This is Advik. He is now 5 years old and he loves the water. Back to science again. Now, I had learned something about life in the ocean itself. So it was time to look at the health of the ocean. But before we talk about that, let's think about why the health of the ocean is important. Say we jump into the ocean and we see no sharks or any big fish for that matter. Is this a sign of a healthy ocean? Now, this actually might be an indicator that there aren't enough predators to keep the herbivores in check, which will either directly or indirectly affect several other levels in a food web in the ocean. So, long story short, I decided to look at predation in Hong Kong. And to do this, I used a tool called the Squid Pop. This essentially refers to a piece of squid, dried squid, that is attached to a metal stake and serves as bait for fish. I deploy them using scuba diving and I leave them there for an hour, after which I return to look at the extent to which the fish have consumed the bait. And that will give me a sense of how healthy the ecosystem is. Another thing that we looked at was decomposition, essentially how quickly and also how effectively microbes are consuming organic matter. These are the scavengers in the ocean, and they also play a very, very important role storing carbon. So, to do this, we used tea bags, the ones that you used to drink, green tea and rooibos tea bags. This is me with my dive buddy Taehoon and also lab mate. So, we are burying tea bags. We leave them there for 3 months in the sediment, after which we come back and then look at how much the microbes have consumed the tea leaves. We simply weigh them and look at the mass loss. And interestingly, we did find sites that have more nitrogen pollution. We found that those sites did have much higher rates of decomposition and poorer rates of carbon storage capacity. Now, coming to mid 2020, I moved to the Bay Area right in the middle of the pandemic. And shortly after that, I had my second baby. This is Adit, and he's a pandemic baby. That's why he has a onesie that says, "My parents did not practice social distancing." He's also a dancer. And so this brings me to the last part of my talk. The pandemic was in full swing, and I happened to be at the right place at the right time. So, owing to my experience with wastewater and also looking at microbes in the environment, I got sucked into some amazing projects. And this was at Stanford University. So, here I'm now going to be spending the last few minutes talking about wastewater based epidemiology. So, to begin, can anyone recognize this person? This is Jon Snow, not from the Game of Thrones. He is a British physician. In August 1854, there was a cholera outbreak that occurred in Soho. At that time, it was assumed that cholera was airborne. But Dr. Snow did not accept this theory. He argued, in fact, that it entered the body through the mouth. And he was convinced that sewage that was contaminating the river was the source. So, after carefully investigating, he plotted the cases indicated in red on a map with pumps, water pumps in the area indicated in blue. And he was able to identify a water pump on Broadwick Street to be the source of the contaminated water. He had the handle of the pump removed, and the cases of cholera began to diminish. This theory, of course, was not accepted until the 1860s, but today he's considered to be one of the founders of epidemiology, which essentially refers to the study of diseases in populations. Now, disease monitoring in wastewater took off since then, and this has been used to gauge aspects of public health like narcotics usage. In fact, in India, the government officials could see the poliovirus re-emerge. They managed to contain the outbreak 90 days before it appeared, because it appeared 90 days before that in wastewater. So this kind of an application has accelerated interest in wastewater-based epidemiology, and the pandemic has continued to do that. Why? Because we see SARS-CoV-2 genes. SARS-CoV-2 is the virus that causes COVID-19, and it's detected in the feces of many individuals. Now, this is amazing because you can track the virus in the wastewater at least 1 to 2 weeks before an infected individual begins to develop symptoms, or he or she approaches a hospital, or perhaps a hospital begins to see a patient as an ICU admission. So, but when the pandemic began, we weren't sure about this. We weren't sure about how to use this tool effectively. So, at Stanford, the team that I was involved in began to look at two big things. The first was, can we correlate the cases that we see in the hospitals to the concentrations we're seeing in the wastewater from large sewer sheds, from huge treatment plants? The answer to this was yes. We looked at sewage treatment plants from the East Coast, the West Coast, as well as the Midwest United States, covering about 9 million people. The next thing that we did was we also realized that smaller workplaces and universities had to close because they could not detect or contain this higher spread of infection. So we looked at sampling wastewater in dormitories, and we realized that it could also be an effective tool to catch cases even when the sewer shed contains fewer individuals than large treatment plants that have millions of residents contributing wastewater into them. So with that, that was the beginning of everything that I did in terms of wastewater. And to end this presentation, I want to talk about what I'm doing right now. At the moment, I'm a postdoctoral researcher at Columbia University. And here I'm looking at not just SARS-CoV-2, but also other pathogens in the wastewater. So imagine if you were in an airport, you can see a dashboard with different flights that helps you get a snapshot of what flights are going where. So the idea is for us to build a dashboard in maybe a university campus and get a snapshot of community health by having a list of different pathogens that are present in the sewer network. So that's what I'm working on right now. And fast forward to 2022, we got a puppy who we fondly call Taco. And I'm very grateful for my wonderful, wonderful family. Of course, this journey has not been problem-free. I've had several hurdles, both personal and professional, but I'm super grateful that the science continued to grow through all this. And I'm here today sharing this journey with all of you. So with that, thank you so much for lending me your ears, and I'm happy to take any questions.
Wonderful. That was so fascinating. I loved hearing your whole journey from when you started all the way back as an engineer and how you got to where you are today. A couple of questions that I've got for you. I'm super curious, just based on what you're working on today with your postdoc. You mentioned that it is kind of could be a snapshot of community health at a campus level, but would that ever then be able to be extrapolated to community health beyond a campus? So for a city or a town or a different type of community?
Thanks for that. Yes, that is the idea, for us to pilot this in a campus and look at a couple of pathogens like SARS-CoV-2, influenza, or maybe some food pathogens, and then see if we could do the same thing across a city. like New York City. And the idea would be, yes, for us to do that not just efficiently, but also quickly.
Mm. And how long do you think that this kind of initial test phase of the project is going to take?
Well, we just got funding for the project. So it's at a very nascent stage right now. And we're gathering resources, potentially recruiting team members to get involved. And we've got initial funding for a year. So we hope to make some progress within the year and then see what to do thereafter.
Exciting. We'll have to stay tuned to see any publications that you guys come out with. I think that'll be really informative and wonderful for on so many different levels. The video that you shared with us about your work in Hong Kong and involving the citizen scientists and the community, like you said, we would see how intrigued everyone was and how engaged they are. How do you go about kind of publicizing or advertising for your citizen science projects? And how many people would you say that you got to help out with that project?
Thanks for that. That was actually one of the fun aspects of my PhD because we got to interact with so many people and they had wonderful questions. So, at first, the government of Hong Kong is extremely cooperative, and we have a very good relationship with the government officials. So, most of the time when we have a project like this, we reach out to them not just for permits, but also to share what we plan to do, and they often circulate this information. They put out posters that they would let the general public know. In addition to that, we also have our own network. So we do advertise within the Department of Biological Sciences, and we have relationships with the Hong Kong International School or other schools. So the teachers would often reach out to us if they would like some students to join and for that effort to be part of some kind of teaching module in their classroom. So the video that you saw is actually involving about, I think, 30 students from the Harbour School, and they're all middle school students.
Oh, that's wonderful. I'm sure it just sparks their interests immediately into what they can do with themselves in the future as well.
Yes,
Um,
exactly.
Kind of along that same lines, all the different sampling techniques that you have. So the hotel, the arms, and then the squid bot and the tea bags, are those things that you and your fellow scientists come up with on your own, or are those traditional sampling techniques that are widely used with everyone, or kind of how do you choose how you sample what you're looking for?
The short answer to that is it's a combination of both. So the arms is something that originated here. I think it was NOAA that came up with the unit itself. At first, it was made of concrete, and then it was worked on, and then Smithsonian Institution took it up to um, come up with a standardized model that could be reproduced perhaps in other factories across the world. So any data that you generate from one arms can be compared with one from an arms somewhere else as well. So the squid pops and the tea bags, the squid pops were also sort of part of like a protocol that we worked with the Smithsonian Institution. The tea bags we did kind of improvise, so it's something that was new and has not been used in the marine environment. So that was something that I worked on and came up with.
It must be so fun to kind of come up with your own inventions of how to sample for exactly the data that you're hoping to collect as well.
Yes, it was used in the tea bags were used by researchers in mangroves and other on land, but not so
Mhm.
much in the ocean. So that was the tricky part.
Yeah. And I know that you kind of mentioned that we don't really talk about wastewater and sewage very often, but how important it is for our earth and for our globe. So what would you kind of say to people about how they can get involved or how to learn more about what we can do to help or what we need to be more aware of?
Oh, thank you for that. Well, I think the first thing to do, I tell my students, is to find out where your sewage goes. Do you know where your pipe goes? For them to just track it. And more often than not, we don't know. whether you have a septic tank at home, is there a treatment plant, or is it just going to the river or the ocean, to just be educated about what happens after you flush the toilet.
Yeah.
And once that happens, I think individuals tend to get more curious about what gets flushed down as well. And you tend to become more aware. So that's the first thing in terms of awareness. Otherwise, I think in general, to be more involved with maybe beach cleanups, or perhaps talk to an NGO nearby or a university, see if you would like to be involved, because we often need help from the public, and the public that actually visit these areas know much more than we do because they've lived there for so many years. So it's just about connecting with your nearest, I think, science group or maybe an organization, yeah, and seeing how you can help.
Awesome. I think that's great. And what a great piece of advice that we should all, I challenge everyone who's watching to go and find out, where does your sewage go? Where does that pipe lead to? And then learn more. Um, Thank you so much. This has been a fast half an hour, but a very, very fascinating one. And so wonderful to hear about your journey and where you are today, and see the personal touches of your family as well. That was wonderful to see. So thank you so much for joining us today at the Global BioFest. And we'll stay tuned to see about a year from now what happens with your current research.
Thank you so much, Lizzie.
Yeah, have a great night.